In nitrogen cycle, we learned that air is a very rich source of nitrogen. The use of microorganisms is one way to make atmospheric nitrogen available to the soil. Let us study this process in detail. In the atmosphere, nitrogen is available in diatomic form. Only a few microbes can fix this nitrogen. These microbes have a special enzyme called nitrogenase, which reduces nitrogen to ammonia. The process by which these living organisms fix atmospheric nitrogen by converting it to ammonia is called biological nitrogen fixation. Nitrogenase is sensitive to oxygen. Even low concentrations of oxygen can make it irreversibly inactive. Microbes that possess this enzyme are called nitrogen fixers. Nitrogen fixers live independently or as symbionts. Examples of free-living aerobic nitrogen fixers are bacteria such as Azotobacter, Clostridium, Klebsiella, and blue-green algae such as Anabaena and Nostoc. The bacteria Rhodospirillum is an anaerobic nitrogen-fixing agent, but it can also exist in aerobic conditions. Nitrogen fixation is slow in free-living microbes due to the energy requirements. Nitrogen reduction needs ATP to supply energy. Some nitrogen-fixing bacteria exist as symbionts. Rhizobium is known for its symbiotic association in the root nodules of leguminous plants such as beans, peanuts, lentils, and so on. Non-leguminous plants such as alder, alnus, ceanothus also associate with frankia in the nodules of their roots for nitrogen fixation. These nodules in cross-section appear red or pink due to the presence of an iron-containing protein called leg hemoglobin. Let us take a look at how rhizobium helps plants. Rhizobium in the soil multiplies around the roots and gets attached to root hair or epidermal cells. Root hairs curl, indicating the presence of bacteria. These bacteria reach the cortical cells through this root hair. Then the bacteria stimulate the inner cortical and pericycle cells to divide. These cells get differentiated as nitrogen-fixing cells, and a nodule is thus formed. Through vascular supply, the nodule is ensured of nutrients and energy from the plant. The enzyme secreted by rhizobium, nitrogenase, is a molybdenum ferrum protein and acts as a catalyst in the conversion of nitrogen to ammonia. As nitrogen is sensitive to oxygen, the nodules are adapted to protect the enzyme from exposure to oxygen. Leg hemoglobin, the iron-containing protein in the root nodule, acts as an oxygen scavenger and prevents its exposure to nitrogenase. Nitrogen requires 8 electrons, 8 protons, and 16 ATP molecules of energy for the formation of 2 molecules of ammonia. Therefore, 1 molecule of ammonia requires 8 molecules of ATP. These 8 ATP molecules are provided by the ATP released during respiration. Let's see how the enzyme nitrogenase converts nitrogen to ammonia. Nitrogenase first binds with substrate nitrogen and reduces it by adding 2 hydrogen atoms during each of the 3 stages until it finally forms 2 molecules of ammonia. After that, the enzyme is re-released to bind to another molecule of nitrogen. In physiological pH conditions of the plant, ammonia molecules take up protons to form ammonium or NH4+ ions that are channelized and used in the synthesis of amino acids. The ammonium ion ultimately forms the amine group of the amino acid. Amino acid synthesis in plants takes place in two ways: reductive amination and transamination. When ammonium ions react with alpha-ketoglutaric acid, it reduces it to an amine and forms glutamic acid and water in the presence of the enzyme glutamate dehydrogenase. Hence, this reaction is called reductive amination. The energy required for this reaction is obtained by the oxidation of NADPH to NADP. Transamination involves the transfer of one amino group from an amino acid to the keto group of a keto acid. The reaction is catalyzed by a family of enzymes called transaminases. This is analogous to a double displacement reaction, where there is an exchange of an amine group of one amino acid with a keto group of a keto acid. This results in the formation of asparagine and glutamine from aspartic acid and glutamic acid. This process takes place due to the replacement of the hydroxyl group of the carboxyl group of aspartic and glutamic acids with the amino group. Amino acids are usually transported through phloem, but compounds such as amides and ureides, owing to a greater nitrogen to carbon ratio, are easily transported through xylem.